thank you all for joining this presentation. My name is Sepehr Jalayer and the topic of our research is VRNConnect, an interactive virtual reality platform for connectomic data exploration. To start with an introduction to our work, brain connectivity can be incredibly complex, and we were motivated to look at the tools we could use to make it easier to visualize and interact with for analysis. Virtual reality has proven to be a versatile tool for learning, exploring, and analyzing complex data. It can expand the user's senses, provide a more detailed and immersive view, and facilitate the analysis of complex data such as brain, which is incredibly complex. It provides an immersive and interactive experience that allows users to feel like they are actually present in the environment. It has enhanced depth perception, allowing users to perceive distance and scale in a more natural way. VR also allows users to interact with objects and environment in 6 degrees of freedom, meaning they can move around and interact with objects the same way as they do with real objects in real life. VR can be cost-effective way to visualize and explore complex data sets, allowing researchers to gain new insights and identify patterns that may be difficult to see with traditional 2D visualization methods. It can be used to simulate environments in a safe and controlled way, doing procedures without putting real patients at risk. Brain connectivity is particularly interesting to study as we can advance our knowledge of both functions as well as dysfunctions of the brain. For example, comparing the connectome of a healthy population with population of people with Parkinson's disease who had broken connections showed that the severity of motor symptoms was linked to how much the network was broken. Similar patterns also can be found for Alzheimer's, multiple sclerosis, and etc. So to give you a bit of background on the subject, the connectome, which is referred to the wiring diagram of the brain, is a map of neuronal connections inside the brain. It is crucial to understand how the brain performs a wide range of cognitive and motor functions. The picture on the left is a sample of brain wiring diagram that can be acquired by means of tract tracing or tractography. The tracts or streamlines represent the white matter pathways that connect different regions of the brain. As for the visualizations used to illustrate and analyze the connectome, some of the most popular ones are the brain surface visualization. Each brain cortical region is represented by a colored patch with different colors representing different connectivity measures. They do not, however, provide information about the brain connectivity patterns in subcortical regions. Connectograms. In connectograms, nodes are clustered in small word networks with high clustering. Connectograms can be used to identify hub regions with strong connections to many other regions, as well as to visualize the overall network structure of the brain. 3D visualizations such as fiber tractography, as we've seen before, allows you to see the anatomical connections between different regions of the brain in 3D space. Network graphs or node link diagrams show regions as nodes and the connections between those regions as edges. Matrix plots, each row and column represent a different part of the brain, and the cells in the matrix represent the strength or directionality of the connection. On the other hand, VR is being increasingly used in the medical domain for medical education, surgical planning, and training. In medical education, some advantages of VR are broadening sensory perception, enhancing the engagement of the students, and etc. Many work have been focused in this area, such as work done by Kyo et al. In 2019 for training healthcare professionals with VR. In VR simulation, a recent review on the usefulness of VR for surgical training has shown that VR can be cost-effective, safe, and successful in surgical training. As for serious games in VR, a more recent development was the game aimed at neuroanatomy teaching by Souza et al., which has shown promising results for both teachers and learners. Overall, the research indicates that immersive experience is an effective tool for medical training and education. And when we focused on all the related work and software that worked with connectome data, we saw that almost none of them provide true immersive VR experiences. And the ones you see in the table that have VR compatibility, They use the VR headset just to project the scene that you view on your typical monitor onto a VR headset. They might give you a better feel of 3D perception, mainly because of the lenses inside the head mount, but they do not provide a truly immersive virtual experience. So, all that being said, now we talk about how we developed VRNConnect. Our system was designed in order to take any connectivity matrix data as input, as well as an atlas file. We provide one, but this one can be overwritten by the user. We use the Unity game engine, and since we chose MetaQuest 2 in order to have the best integration with its latest features, we added the Oculus Integration SDK. We also use Python version 3 to handle intermediate data creation, calculate the graph network measurements, and connect to the Brain Connectivity Toolbox library to provide the needed matrix. To visualize the connectome data, we use a 3D graph with nodes and edges where the color coding of the nodes is obtained from HCP-MMP1 atlas. Edges connecting those nodes are visualized as gradients of the two nodes they interconnect. Weights of the connection is visualized by adjusting the thickness of the edges. In addition, a semi-transparent brain mesh created from a segmentation of MR images is also visualized for greater anatomical context. Moving on to the interactions, we used Oculus Integration SDK to have better integration with Oculus touch controllers and gesture detection. As for gesture detection, the Oculus headset, we decided to implement the pinch gesture for now. As you see in the illustration here, we have the base move and camera rotation interactions implemented on the controller's input buttons. And with the trigger button, you can select, deselect, which can also be done with the pinch gesture. Regarding the interface that we developed for VRNConnect, we added two canvases, one for showing the regional information regarding a node when a specific node is selected using either hands or controllers, and the other canvas is for changing visualization settings. We have the toggle to show or hide edges, the threshold slider to change the upper bound of the thresholding we did on the connectivity matrix, the scale slider to change the whole scale of the brain for better viewing the dense areas. Path algorithm is a drop down with two options, hops and distance, which changes the way we calculate the shortest path between two selected nodes. In order to evaluate our system, we conducted a short user study. Participants undergo an oral introduction of the system, then they fill a pre-test questionnaire with mostly demographic questions, followed by 18 steps of interactive tutorial inside VR, making them comfortable and familiar with our application. And immediately after the user study, they fill out the post-test questionnaires SUS and TLX. Two different tasks were designed for, and for each task, 3 sets of nodes were to interact with. Once with controller and another time with hand gestures. The order was randomized for each user. In total, 12 tasks for each participant. Task 1 being the node selection, you can see in the left column. Task 2, shortest path, you can see in the right column. And during all these trials, time and the number of clicks were recorded in order to have better understanding and interpretation of the results. Looking at the results, we had 16 participants, 8 male, 8 female, with average age of 28. 62% of them had little to no experience with VR, and 75% had little to no knowledge about the role of the brain connectome. As for the time of the task completion, we can see that it tends to take almost the same time for participants to do both tasks, which can point to the fact that although task 2 were required more interactions, but as they got familiar with the system, they started to do things faster. Also, regarding the comparison between hands and controllers, as we expected, the controller, it took far less time to complete the same task as doing it with hands. As for the system usability, which is a 10 item questionnaire with a Likert scale, it records the subjective perception of the usability and user friendliness. The mean score of VRNConnect was 86.25, which according to Bangor et al. And Suara et al. Is considered excellent. You can see the average score for each item in the table. To conclude our research, In a nutshell, the contribution of our work were development of a virtual reality platform for visualization and analysis of brain connectivity data using MetaQuest 2. The flexibility of input, although we used structural connectivity data for evaluating our system, users have the versatility of importing their own data, whether functional connectivity or structural. Intuitive interaction using both controllers and hand gestures, allowing users for better understanding and convenience of using either hands or controllers. Development of a Python script running in the background, allowing for connection to use brain connectivity analysis tools during runtime. Dynamic display of graph network measurements such as node strength, degree, and clustering coefficient alongside the regional information of the nodes in VR environment. Dynamic calculation of and visualization of the shortest path between two nodes. Evaluation of the VRNConnect features and interactions using both hands and controllers in terms of usability and workload. Thank you all for listening.